So, tuberculosis is a serious health challenge, and government of India wants to eradicate TB in next 5 to 10 years time period. It's known to alter the human immune responses once it infects our body, and our group has been trying to understand what are the mechanisms through which bacteria alters host immune responses. So one of the fundamental way through which host response happens is by changes in gene expression, and gene expression in humans and all eukaryotes happens through a set of something called disrupted genes, which are introns and exons together, and they have to be spliced and stitched together to form a functional messenger RNA, which can be translated to form a functional protein. So what we have reported is that tuberculosis bacteria, which is Mycobacterium tuberculosis, when it infects the human cells, specifically macrophages, which is the immune cell, it alters the way this stitching and splicing of messenger RNA happens inside the cell. So in a recent study, what we have shown, we have come up with a mechanism that how Mycobacterium tuberculosis secretes a virulent protein, and that protein actually alters the protein complex which is involved in this stitching and splicing of messenger RNA, and that completely blunts the host response against tuberculosis, and that helps the bacteria. The interesting aspect of it is, since we know now the mechanism, you can try to come up with strategies to overcome how to inhibit these bacterial virulence functions and strengthen the host responses to control bacterial survival and pathogenesis inside the human host.
This is a huge effort, as is evident from the list of authors that you see in the paper. A lot of people have worked together in a combined effort to make it a success. There are a lot of people have worked hard to see it through and make it a success.
Ours had been a very diverse kind of team, comprising from basic biologists to clinicians, from basic molecular biologists and cellular biologists to translational researchers. Our paper has recently gone online, and it can be accessed in the July edition of the Proceedings of the National Academy of Sciences, or PNAS journal.
We uncovered a new mechanism deployed by MTB to thrive effectively in our body. It releases a special virulence protein that interacts with the RNA splicing machinery in the host. This machinery is important to interpret the genetic information and make functionally important proteins for our survival. Therefore, when the protein interacting with this machinery is removed from bacteria, its survival is compromised.